Bioplas, dependable solutions for biotechnology and the clinical laboratory. Bioplas is proud to introduce new products: the patented Astral disposable plastic inoculation loop system, the Astral streaking needle, and the Astral clone pick. All display an impressive collection of features: accuracy, convenience of use, loops calibrated using both the Evans blue dye spectrophotometric procedure as well as a gravimetric method using Remel nutrient broth, time-saving, economical, dependable sterility, versatile. They may be used in lab settings and in fume hoods, or even in the field with maximum protection from contamination. Certified free of DNA and PCR inhibitors. Of particular importance for the Astral clone pick when used to inoculate the polymerase chain reaction for rapid characterization of DNAs cloned in various vectors. Packaged free of static electricity. This presentation describes the Astral disposable inoculation loop system, the Astral streaking needle, and the Astral clone pick by Bioplas, and their uses in modern biotechnology and clinical labs with the following applications in mind: pipetter independent dilution series, semi-quantitative re-inoculation of liquid microbial cultures, direct inoculation of the polymerase chain reaction, or PCR, with plaques or colonies for rapid characterization of DNAs cloned in viral, bacterial, or yeast vectors. Using the Evans blue dye colorimetric procedure, the Bioplas Astral inoculation system falls well within the guidelines recommended by the Clinical Microbiology Procedures Handbook, published by the American Society for Microbiology. To further assess the accuracy of the Bioplas Astral loops, we tested these loops with Evans blue dye and the Remel nutrient broth using the gravimetric or weight method. The scale used in this test was a high-end Mettler of the AG series. The results from using the gravimetric method also fell within the guidelines recommended by the Clinical Microbiology Procedures Handbook, published by the ASM. The development of the Astral inoculation system was designed not only to obviate an inaccuracy of contemporary methods, but also to enhance the laboratorian's efficiency expended on any specimen, whether it be for subculture or quantification. All Astral system products are packaged in a very sturdy 96-well rack. These racks have 96 tubular chambers open at the top. The tubular chambers have a molded shelf that holds the Astral product upright so that they may be easily removed. After the 96 well racks are filled with Astral products, a very thin adhesive aluminum foil is sealed over the entire 96 well rack. A dust cover is then placed over the entire top of the rack, heat shrunk, and then 10 racks are placed into a cardboard carton. All this packaging is performed in a clean room. The product is then sterilized by electron beam sterilization, designed specifically for medical devices in their final shipping containers. Astral disposable plastic inoculation loops, the Astral streaking needle, and the Astral clone pick are further verified free of DNA, RNase, DNase, pyrogens, and PCR inhibitors. The adhesive aluminum seal is easily punctured with a loop handle and offers ready access toward rapid removal of the sterile product from its compartment. Each case of sterilized Astral inoculation loops, Astral streaking needles, and Astral clone picks are supplied with an Astral handle, which is used to withdraw the Astral product out of the wells. This plastic Astral handle is 6 inches long, with a movable sharp pin on the bottom tip of the handle. It has a flat portion at the top to be used to eject the loop off the handle after inoculation. Once the Astral inoculation loop, Astral streaking needle, or Astral clone pick is secured to the Astral handle, they are ready for use. Either a 1 microliter or 10 microliter loop may be used to inoculate either a sterile prepared petri dish or tube. The Astral handle may be autoclaved repeatedly if needed. The clone pick by Bioplas is expected to support efficient screening of DNA inserts in cloning vectors. Rapid examination of DNA clones is now routinely done by amplification with a polymerase chain reaction, or PCR, followed by automatic sequencing. DNA clones in viral, bacterial, or yeast hosts contained in appropriate recombinant vectors are simply picked from selective agar and transferred directly into the PCR mix. However, reports warn of inhibition to the PCR from more conventional devices, such as wooden toothpicks. BioPlas takes the worry out of your work by offering the reliability of the clone pick. This simple plastic device, preloaded in boxes of 96, is designed for manual picking and transfer of colonies. Representative lots of the clone pick are tested for absence of DNA and PCR inhibitors. And rest assured, they are also free of DNase and RNase. Clinical and biotechnology laboratories will find the astral streaking needle a most useful alternative to the toothpick for microbial cell purification on semi-solid agar plates. The astral streaking needle has an end that is adequate for precise and discrete access to single colonies, yet with a smoothness at its end that diminishes the risk of unacceptable scratching of the agar surface during restreaking on the fresh plate. The flexibility of the plastic needle is another feature of the astral streaking needle that laboratory workers often miss in the more rigid conventional toothpick. The compliance of the needle to slight pressure offers more control on the trajectory of the streak as it travels on the surface of the agar. The astral inoculation loop system has 2 patents issued. All Astral products can be ordered from the following BioPlas authorized distributor or by calling BioPlas at 415-668-8917. BioPlas, dependable solutions for biotechnology and the clinical laboratory.